Hey, folks, we're setting up again. I don't know why Judith and I always have this problem, but she's out in rural New York and having terrible connections. So I just don't know what's going on. Come on, Judith, let's go. Hey, South America, we're translating the books in Spanish as we speak. I'll answer a couple of little questions here. Hi, Adam, how you doing? Hey, Gorilla Farms. Yeah, kind of weird. This stuff is weird. It's got something to do maybe with the elections or something. I don't know. Let me try Judith again. Let's see here. Just sent out a new invite to Judith. We'll see what happens. And we're waiting for her to come. Well, see, Johnny. Yes, nice to see. Oh, Judith is connecting.
Yeah.
Hey, we did it.
Yeah.
All right, and it's 11:58. We still have a couple of more minutes to go, which is very good. So we're going to start in a minute. How you doing, Judith?
I'm doing very well. How are you doing?
Now I'm doing.
And thank you both for coming in. It's really nice to meet with you guys again. We're really loving the input you're giving us.
Good. And how are sales this week for the microbiometer?
Well, we're doing a lot of work with Terraton now.
Okay.
On carbon sequestration. And I mean, one of the horses I'm on is right now is the fact that a lot of the carbon sequestration people, I don't know if our group is interested in that, but they keep emphasizing, putting carbon back in the soil, and that they're going to give people like $15 an acre to do that if they can guarantee it for 100 years.
Yeah.
Which,
Yeah. But you can't just put carbon in the soil.
And a lot of paperwork and everything, and they really don't discuss yield
by adding carbon.
But for every grower, the most real thing is your yield, right?
Absolutely. Unless you're growing, no, for every grower, you're absolutely right.
I mean, we're growers, so that's what we want is growing. Right. But also, I got to brag a little bit.
Okay.
Because I'm in New Jersey.
Yes.
And cannabis just became legal in New Jersey.
Congratulations. So what does that mean? It means you're going to get to start growing plants legally. Wonderful.
Yes,
Good. Well, we're going to have to make sure that they sell lots of auto-flowering seeds there, because that's clearly the way to grow in New Jersey. You want to be able to grow all winter. You want to have more than one crop. That's the way to grow. So what do you want to talk about today? You want to talk about carbon sequestration and aggregate formation. Is that correct?
Well, yeah, I wanted to talk about, a lot of people ask us, why do they see variation in the microbiometer test?
You mean using the same soil, they'll take the same test and get a little variation?
They see some variation.
Okay.
And what most people don't realize is that when they send their test to a lab, if they would send the same sample to a lab, they would see the same type of variation.
Of course.
If they would send duplicates.
Yes.
Yes. In fact, James, who was one of our founders, James Sotillo, your friend of mine who died, he wanted to run these tests himself because he was getting 50% difference, at least, when he would send duplicates to the lab in Washington. Elaine comes with it. So we don't get anywhere near that amount of variation because we measure more. But I wanted to kind of, go over why one actually sees differences, and that's because of soil aggregation, as you pointed out.
Okay, so let's define what soil aggregation is. Soil.
I'll leave that to you.
Okay, well, soil is supposed to have great structure, and the structure is formed by the microbiology in the soil, and that structure results in little teeny particles of soil which stick to other particles of soil as a result of a couple of things: bacterial slime, which gets on one particle and makes it stick to another particle, and fungi, which will weave through those various particles. And one in particular fungi that is so important, as we've talked constantly, are the glomalin producing mycorrhizal fungi. And so all of these things work together along with other members of the soil food web that weave themselves through these aggregates as they chase each other and breed and look for food and store food and whatnot. And that forms these wonderful aggregates of soil, which when looked at are actually like irregular things that are just stuck together. So there's lots of pore spaces, air spaces, water can be held in, little hiding places. These are not bricks that are put together. Now, when you rototill too much, when you use too much chemical fertilizer and reduce the microbiology in the soil, particularly the fungi, I might add, because the fungi are producing glomalin in particular, we can talk about that later. When you reduce the microbiology in the soil, when you impact negatively the soil food web, you end up with no aggregates. You've just got these fine little particles. So if you've ever seen a farmer who's rototilled like crazy, and then it rains, and the soil turns to what? It turns to mud, and then that mud turns to cement. It's because it doesn't have these soil aggregates. It has no soil structure. Okay, so that's what soil structure is, and it's formed by the soil food web, and you can learn about it in Teaming with Microbes and, of course, Teaming with Fungi. I turn to you now.
Okay. So, in a lot of ways, I like to think of it as being lots of little caves.
Excellent.
And these little caves are homes. So, in a way, you're thinking like these microbes are making little homes for themselves.
Right.
Okay.
Little condominiums.
And in the way like little fish in a stream go hide under rocks and things like that, these microbes can elude, the people who are the other microbes that are chasing them for meal.
Right, exactly.
By hiding it. And also, they need a place to attach. So they don't want to attach like on a main road. They want to attach, on a side street, so to speak, in a little place like that. Right. And then these little caves become spaces for holding water and holding air. So you're not holding water and you're not holding air if you don't do that.
Well, they also have an electrical charge, and they hold nutrients as well. So some of them.
Exactly. So, if you take a half cc of soil and run it in our test, okay, you're releasing often close to 1 billion microbes.
Right.
And, if you look at these aggregates when they're there, one of these little caves might hold 10 aggregated together. Sure. So there's lots and lots of them. And so I think of it like this. Think of apartment houses in New York City. So I just took a whole bunch of apartment houses in New York City. I took apartment houses in New York City that are holding 1 billion people.
Exactly.
Okay.
Sure.
And now I am going to measure 10 of them.
Not so easy. Yeah.
And I'm going to say how many people are in those 10 apartments. So if there's 1,000 people in an apartment, I'm looking at what? 10,000 apartment building, sure. 10,000 tests. Yeah. So every time I take a sample, I'm not going to find this, even if the apartments are all exactly the same, I'm never going to find the same amount of people in those apartments.
That's a nice way to put it.
So you're just not going to get exactly the same. But when people say, and the CVs on like nitrogen in soil, it's not uncommon at all for those CVs to be 25% in the lab when they're measured, from two from the same sample. Sure. and so there is a lot of variation that is just intrinsic to measuring a biological system like this, when you have to measure just a portion of
Sure.
it, not a huge swath of
Sure.
it.
But you get a base, though. I mean, in that condominium, there's a base level that you're able to measure. So that the number you get is a valid number. It just can vary depending on what floor you're testing on. Maybe that's
It can vary. Like you're gonna usually you can easily say, like, we get a 10% CB. The 10% CB is probably just based on how much of the soil we actually measure.
Sure. Okay.
If we would measure more of the soil, just like if you're doing, you know, a presidential, looking at a bunch of people to predict the outcome of an election.
Sure.
The more people you have, the more accurate you become.
Hopefully.
Okay.
That's right.
So the more microbes you can measure, the more accurate you become. And this is true throughout all of laboratory testing.
Right.
So, we validated against microscopy. And what we see with microscopy is we can't bring the CB down to under 25%.
Yeah.
So there's 25% variation because we're measuring like 1/1000th of the number of microbes that we're measuring with microbiometer.
Well, and also aren't we using a visual, a human being as opposed to artificial intelligence, which the microbiometer.
We're using
Brady
developed artificial.
Yeah, you're using artificial.
Brady.
Right, but the
But we validated against digitized microscopy,
right,
which measured everything by calculating from 30 photographs of full fields, counting each microbe in there and calculating its volume.
Right, and I guess my point was that what you used there to match was a digitized system. When you send out your samples to be tested by a lab, most often it's an operator that has a grid, and that operator is counting individual organisms through a microscope. And so depending on the operator, depending on what the operator had for lunch, depending on lots of different factors, that operator can come up with a different reading. So all of this is to say that because of those wonderful aggregates, and because of the way Brady has developed your system, we get a much more accurate reading than we would if we were using an eye system and one that doesn't employ artificial intelligence. And which ones of these other systems do employ artificial intelligence? I don't know any of them that do.
Okay, yeah. I mean, we know that if you use a system like that, like Pap smears used to be done by eye, and they were not nearly as accurate as now they're done by digitized microscopy.
Right.
So we didn't invent digitized microscopy or anything, but we applied it and developed it for soil, and we did validate the fungal to bacterial that way by identifying fungi by their shape and their size for digitized microscopy.
Right. Which is
and microbes.
And that's got to be pretty complicated, but Brady nailed it, no question about it. I mean, you guys have. Whew.
Yes, it's very complicated.
What a system.
We measure like 3 different slides, 10. We have a pattern. We go around and do 10 areas on each one, and then there's usually 3, 4 to 100 to 1,000 microbes that are counted on each picture that's taken.
Wow.
So each and it's done on our extraction fluid.
Wow. Well, let me ask you the idea that these aggregates have an impact. So, because I know I've tested soil that's been ground up into a fine powder versus soil that has an aggregate. If you do it both at the same time, you get the same reading. But if you take your aggregate reading a month after your powder reading, you get a difference. because the microbiology is growing and living in those aggregate spaces, et cetera, et cetera. And
Yeah.
I don't think
Yeah.
and I don't think people appreciate soil structure enough. It's the be all and end all. If you're not building soil structure, you're really not adding to your soil. I don't think. That glomalin is where the carbon comes from that goes into our soil. Most of it. The other thing is that the residues that would normally be on the ground after a plant dies, those residues protect the soil structure. You get rain, you get snow, you get that stuff gets buried, it gets taken in by a worm. They're spongy. They retain their shape, and so they help the soil structure maintain aeration, etc. Whereas if they were ground up and pulverized by a rototiller, or not there at all, you don't have that spongy material on the surface and being brought into the soil itself to keep and protect that soil structure. All of this stuff is
from rain and sun, because it gives temperature protection.
Sure, or even farm equipment,
protection.
even driving over it with a tractor. You drive over your plant with a tractor, it bounces back. You drive over ground up plant with a tractor and you make a fine powder cement. And so it makes a big difference. But again, that glomalin, we can't stress enough how important that fungi is.
Well, I've been thinking about you because I was reading a nice paper this week.
Oh.
And the paper was saying that roots that are left in the soil are actually far more important for building soil organic matter than the foliage that comes down. And I'm sure that part of that is because the roots have a lot of arbuscular mycorrhizal fungi.
Absolutely.
In a lot of cases.
Absolutely.
So they're not only bringing all that organic matter there, okay, but they're actually a lot of glomalin that's with it, and then they're staying there to
food source.
Connect with the next plants that come along, because as soon as the plant starts to die, these AMF go into spores.
Right, and then they're attracted by the new plant.
Of course, when I go into a vineyard in the winter, I get these huge readings, and when I look at it, it's all spores, because the vineyards are just loaded with fungi.
Right. Now, of course, it depends on what the crop is, of course, but you're absolutely right. And if you don't, again, the whole idea of glomalin in maintaining and using that as sort of a fertilizer, it's a soil builder, and you've got to have it. You have to have it. So when we talk about fungal bacteria ratio, if you just have bacteria, that's not good. And this is one of the reasons why. And that's why the microbiometer, taking a fungal bacteria ratio reading, gives you a little bit more confidence, or takes your confidence away with regard to your plant's ability to be able to attract those fungi, and for those fungi to be making carbon for your soil. It's so important, just so important. So
right.
when you are doing, by the way, I wanted to ask, where's the furthest away place you sold the test kit to this week?
I don't know, where we have we sell quite a bit in Australia, and I think we actually have an Australian guy watching this today from Australia. So we have a very active sales in Australia, and now we're doing quite a bit in Chile and Peru.
Right. Yeah. Oh, great.
And throughout South America, we have a lot of interest in third world countries, because they really can't afford the kind of soil testing that goes on. And we we've just been working, we worked with the University of Tennessee, and we just finished a study with Cornell. And what we've been able to show is that we are correlating with yield, and we are correlating with soil health.
No question.
Okay.
No question.
And we have a new study going on with Cornell, which I'm very excited about. And they're just working with us, Janice Tice, who's very well-known author on soil health. So
yeah, things are going great.
It's exciting to see what people are doing, and it's good for me to see people saying, the first thing we really have to do, and the really big reason we should be regenerating our soil for everybody, is to feed the people of the world, because what's happening in Africa is due to climate change,
right,
etc.
Right, and
I want you to address a question.
Sure.
I think may be interesting a lot of our people on this. And I know Brian Waxman at one time had said he doesn't like to just throw his soil out, like a lot of people buy new soil all the time. And he likes to keep it as long as it's healthy. Now, if he had a plant go bad, he throws it out and everything.
I agree.
And that makes so much, what?
I agree.
It makes so much sense, because you've added all that organic matter to it, and you have all the microbes there that are the ones that were encouraged by a healthy plant.
Right.
And why would you want to get rid of that population?
Well, it's not only that.
So I'm asking you.
Yeah, no, I agree 100%. I mean, it's always been my philosophy. You keep the soil, you reuse the soil, just if for no other reason what we've been talking about today. The soil aggregates have been formed. They're there. All of those wonderful bacteria glues have stuck stuff together. You've got glomalin sticking stuff together. It's just and it's there. It's been prepared. Soil doesn't get prepared in just a nanosecond. It takes a while, and it's like fine wine. It gets better and better and better. Now, you're right. At some point in time, you can really have soil that gets messed up. You can get mites in it. You can get it compacted, and you need to repair it. But even then, I keep the soil. There's no reason to ever throw soil away, particularly if you're a soil food webby and you've been taking care of your soil and you've been adding compost and organic inputs to it. For the same reason that we plant in existing pots when we grow cannabis, that pot has it's just prepared itself. It grew a spectacular plant. That plant left all sorts of terrific residues. You're going to throw that stuff away? Craziness, absolutely craziness. And then you add into the fact that it takes a while for the soil food web to absorb the nutrients and hold them in their bodies. So when you feed the soil, it takes a while for that nutrient material to get into the soil food web. We want it in the soil food web because that's what holds it in the soil so it doesn't wash away. Whew. Man, you're going to just throw away and put in new soil so that you have to rebuild up the whole system. It just makes no sense whatsoever. And time again, and people here, I've been looking at some of these comments here, people who don't rototill end up with higher yields, which is what we talked about earlier. And that's what it's all about, getting higher yields, whether it's cannabis, whether it's food, you want higher yields. Not rototilling preserves the aggregates, to bring us back to where we were. Preserving the aggregates makes sure that you're enabling those wonderful spores to sporulate and do all the things they need to do. So yeah, it's all tied together. It's just absolutely beautiful stuff. Now I see here, Judith, that we got a couple of people have asked some questions about whether what they're doing is going to ruin this. Somebody asked about using hydrogen peroxide, which a lot of people do. When you take a seed, you want to pop a cannabis seed, for example, you might throw it in water. A lot of people put in a couple of drops of hydrogen peroxide. And why do they do that? They do that because it kills off the bacteria. Now, would you put hydrogen peroxide on your soil for any reason? I wouldn't. So that's something that people do. They use hydrogen peroxide, food grade hydrogen peroxide in the soil. I don't think it makes any sense. It might make a little sense when you're propagating a seed if you want no bacteria in your water, but it doesn't make any sense in the soil. So that's one question I got here. Let's see here. We got another question here about, let's see here. Wait a minute. A couple of election comments. Oh, yeah, here. Someone said third world country. You mentioned third world country. Aren't we a third world country right now? Okay, I won't get into the yes, hydrogen peroxide will kill off specific things, and I don't want to discourage you from using it, but understand what it does. If you use hydrogen peroxide to kill your mites or to kill your fungus gnats, you need to go back and reestablish the populations that you've destroyed. What populations have you destroyed? You could have destroyed an awful high number of them, no question about it. So you got to be careful about what you do, no question about it. Now, here's a question, Judith.
Well, another question I want to ask you about or discuss with you today is a question we get from some of our customers, and they send in soil from a field they just made organic, and as well as soil from a field that's not organic.
Right.
And then they say when they get the results, how can it be that that the organic field doesn't have a much higher microbial count than the field that they've been doing rather conventionally? And sometimes even the organic field is lower.
The question is,
and the issue is, of course, that you have to do, just because you say it's organic doesn't mean you've given them everything they need.
Sure, I was going to say there's two items here. Yeah, being organic doesn't necessarily mean that you're going to have the best yield. You want to have the best organic. You want to have organic that has the right balance between different chemicals. You want to have organic that has the right pH. I mean, you can have organic with a terrible pH and not get any yields whatsoever. And the reason why you might have more microbes in the non-organic is because a lot of these things that we feed the system attract microbes, but they may not be the right microbes. And so you end up with either a lack of diversity, or you don't have the proper ratios so that they're impacting each other the way that they need to. So yeah, you can have completely much higher readings in the non-organic, and by the same token, you can have lower readings in organic and have very, very good yields because you've got the right stuff. Make sense?
Well, I mean, I always loved Kirkby and the new paper by Coonan and all of them, talks about the fact that the fresh organic matter, which is what the microbes are actually using, needs to have, a very specific ratio of carbon to nitrogen, to phosphorus, to sulfur. Okay.
Right.
And if you can't, a lot of times when people put down green manure, the green manure may not have enough nitrogen. So if you're starving your soil of nitrogen or of any other mineral that's required by the plant, you're not going to see, the kind of yield you want, and you're not going to be able to grow your microbes, because just like us, without a vitamin, without one of these things that it needs, it's not going to go. So you should be using microbiometer or some other test to tell you how many microbes you have, to actually see if what you're doing organically is actually growing the population, because what the microbes need is exactly the same ratios that the plant needs.
Right.
That's why the plant grows them up.
Yep.
Yep.
I mean,
so
and the pH, of course, has an impact as well, because the pH can cause things to become locked up chemically. We don't want to do too much chemistry, but that is a big factor as well. So one of the questions that I always get asked by people that you can help with is, so you run out of here's, and it's specific here. Someone's just asked it. When you run out of carbohydrates in your soil, which means your carbon is low. The microorganisms go dormant. Do any of them die because they don't get food? And how does the microbiometer distinguish, and is it important to distinguish between dormant and living microorganisms? A lot in that question.
Wow, that's a book.
So let's start with the microbiometer itself. Can it distinguish between live and dormant microorganisms?
Well, I suppose, it doesn't distinguish between them.
Right.
We do see differences in size. And when I see soil, like in winter, that's really quite dormant, the microbes are like smaller. And I listened to a wonderful talk at the last ASM meeting where the woman talked about the fact that dormant microbes, even if they don't have capsules, their slime interacts with clay, and they actually encapsulate themselves in clay.
So they form.
I mean, what we notice
that's a zygote that it forms. It's a little.
It forms like a spore.
Yeah, I think
it basically is a spore. Like it doesn't. It can't make a spore, but the clay particles interacting with that make a hard crust on it.
Right.
So, so it's very resistant, and they can live thousands of years.
And they can stay dormant
that way.
Yeah. Okay. Yeah.
But they're at different. I think of it like an orchestra. This division comes to life when the plant, which is the director, gives them the sign.
Right.
So they're never going to be all active at exactly the same time. The plant is actually making all the calls. It's saying what you're going to be doing. And so you need those dormant microbes. And a lot of them, it's a system that has been in existence now for 3 billion years.
Okay. Yeah, we're not going to improve on it much.
What?
We're not going to improve on it much, I don't think.
Right. So, we know that different ones make it's a real society, and it's very like an economic society. And different microbes make different components that are needed by different microbes. So you bring one microbe in, and it makes vitamin B, for example, and there's another microbe here that can't really wake up until someone gives it vitamin B. So, because they have very small genomes.
Yep.
They can't make all the things we can. We actually can't make everything we need. There are certain vitamins we have to get. I mean,
we use the bacteria to do it. Right.
These guys just have a small amount of DNA, and they're very reliant on each other and the community in order to survive. And it plays like an orchestra. I mean, it's an astounding thing. And they have within a species, 25% difference in their DNA. So they pick up and they lose DNA as they need it.
Right.
And that's one of the reasons I think, with AMF, that a lot of people are saying you're really better off raising your own AMF, because AMF is so specific to different types of soils and different things. Not that you shouldn't start with something, but if you bought AMF and you put it in your soil with your plant, that AMF has probably changed quite a bit during the course of a year while it's been nurturing your plant. And now it's really become acclimated to your pot. That's why one of the reasons I would say that makes your soil there very valuable.
Interesting. And when we talk about the dormant, that's different from the dead. I think we've talked before, there is now a very strong theory that humus is simply dead microbes that have been taken out of that dormant stage, broken down as much as they possibly can. And what's ever left and can't be broken down anymore is humus. And so the idea that you have dead microbes is sort of, you got to really think about it a little differently than normal death. Dead microbes turn into something really phenomenally good, and having a lot of dead microbes, if they're the right ones, can be a terrific thing. Let's see here.
And they attach to the soil particles too. So if you don't have any soil structure, it's going to be hard to hold on to your organic matter.
Exactly. Hey, I see we've done our half hour here, but we continually get this question, and I just see it came again, about adding terpenes to the soil in order to get more terpenes in your plants. My take on this is as follows. I want to see whether you have a different take. What you put into the soil feeds your microbes. Your microbes then produce the individual nutrients, the 18 nutrients that your plants need in order to live. And that's the role of those microbes. And if you're putting terpenes in your soil, what's happening is the microbes aren't taking those terpenes and converting those terpenes into a terpene form that the plant can use. The microbes are breaking those terpenes down into their individual components, putting a charge on those components so those individual components can go into the plant. Once in the plant puts together the terpenes that it needs in order to protect itself or do whatever it wants to do with those terpenes. So adding terpenes to the soil might have an indirect increase impact on terpenes, in that it makes available the individual nutrients that go into making up terpenes. But I don't think it helps. I know it doesn't help by adding terpenes that end up becoming plant terpenes. Does that make any sense to you?
Well, it does actually. I mean, you don't build up protein in human beings, but you do it by giving them proteins, but actually those proteins are broken down by bacteria in the gut.
Exactly.
And then you're absorbing amino acids and things like that. Exactly. So it's food.
Right. And I think people have to understand that.
That's what you're saying.
It's food, and it's good food, and it's got all of the necessary things in order for the plant to be able to make terpenes, but it itself may not necessarily be terpene producing. And the reason why this is important is because there are instances where you don't have the right nutrients and balances of nutrients for your plant to be able to make good terpenes. So adding those can be very helpful. But there may be lots of situations where you've already got everything that your plant needs in order for it to make those terpenes that you want to have in your plant. So I think you get...
Well, the other thing to remember is that it's the low-level stress that organics creates, where, they're just living a normal life, okay, that actually makes these plants make the terpenes, because they're part of the defense system of the plant.
Exactly. And when people think about terpenes, they need to understand that the terpenes in your cannabis plant, at least, forget the terpenes in your grape and your orange and everything else, are in those trichomes. And those trichomes are only 2 or 3 little molecules. They're filled with a liquid that's got the stuff in it that makes the terpenes. These are little teeny cells. And they didn't take the terpene up from the soil. They took the nutrients up in order to make those terpenes. Anyway, you probably need to go make dinner, don't you?
Yep, I guess so. Yeah. I guess so. And my phone is getting low. Okay. So thank you so much, my friend.
Absolutely.
I'm watching television these days.
Really, it's so nice to be able to talk to somebody. I think my spouse must be getting Judith must be getting so sick of hearing my voice. But it's so nice to be able to talk to somebody that, who can say, oh, okay. Anyway, a lot of fun, and we'll do this again. And I think there's a way to save these things so that people can keep them.
Oh, yes.
Let's see. We had what are the longest?
Well,
the two quick questions.
We have Brady and Laura.
Okay, so we got a couple of questions somebody asked, which I'll try to answer if I can quickly. If you need to hang up, one of them is, what's the longest living microbe? I don't know, but again, I think our discussion that microbes can live forever, is sort of
Sure. If this person says, if I have a better pot that's been producing great plants, can I use some of it to jumpstart new pots or beds?
Sure, absolutely.
Should be good.
Yep, absolutely.
That folks are recommending.
I love it. All right, folks, we're going to do this again. We're having a lot of fun.
Good night, everybody. Thank you so much for joining us.
Absolutely. And stay well and mask up, folks. We're going to beat this pandemic. We're moving forward. Talk to you soon, everybody.
Bye.